My name is Patrick. I'm one of the software engineers who works on the Hail team. So for the second practical, our goal is to work through some simple genome-wide association tests. And I want to say up front, I'm a software engineer. I'm not a geneticist. So I'm going to do my best to motivate the steps and explain what's going on. But some of you probably know better than I do. And feel free to interrupt me with questions, or if you want to add any explanation. Okay, so like we did in the first. Okay. Like in the first one, we need to start by importing Hail and setting up the plotting, and then initializing Hail and telling Boca to draw plots in the notebook, not to files. Okay, and then with that ready, we can read. So in the first one, we imported files from different formats. In this case, we have a table that's already written in the Hail matrix table format. So we use the method read matrix table to read that in and make it ready for use. And so before we go on, so at the end of the first practical, there were some questions about how do you sort of see what are the different methods available. And so there's a couple things you can do. So there's a general Python function called dir. And so if we run that, we get just a complete list of absolutely every method. And all these things with attributes, or sorry, all these things with underscores are considered sort of private in Python, so you can just ignore those. But then here's a list of the methods that are meant for you to use. So that's the sort of complete raw list. But maybe more useful is if you do a dot and then tab, and then you got the autocomplete list. And then another nice trick is, so if you complete one of these and then add a question mark and run that, you get a little pop-up here with the documentation for that method. So this is maybe a useful way to sort of explore what's available in the interface. You can also use this on the object itself, so the matrix table, and you get the documentation for matrix tables. Okay, so let's keep going then. So the first step in a GWAS is typically we want to get rid of all the really rare variants. So the variants that appear in only, say, less than 1% of the population, because there's just not enough signal there to see an association. And one thing to notice about the matrix table interface is calling filter rows doesn't change this matrix table we're using. It's actually giving us back a new matrix table. And so we have to say, so this is a common pattern of mt equals mt.filter rows. So that means filter rows and then store the result in the same variable mt. If I wanted to give it a new name like filtered, I could do that. And then I would have MT from before I filtered and MT filtered from after I filtered, and you would still be able to use both of those. Okay, so now, as we were doing in the first practical, it's a good habit. Oh yeah, question.
Are these per each row is like per variant, or all the variant for that loci? Is it like multi-variant or?
The filter?
Yeah, so here like AF bracket 1, right? So is it for one variant or multiple variants?
The matrix table has multiple variants.
Per row.
Each row of the matrix table is a variant. So if we look at the picture here, each one of these is a variant.
And so the question's about biallelics versus multiallelics?
Yeah,
right?
multiallelic.
I think in this case, this data set is entirely biallelic. There are functions to put multi-allelics into biallelics, or you can adapt the code you're writing to work with multi-allelics. But things get a little more complicated in that world.
OK, so it's a good habit to use describe to just get an overview of what are all the fields, what's all the data in this matrix table. So like before, the rows are keyed by locus and allele. And then we have some other summary information about each variant. And then the columns are keyed by the sample string. And then we also have some phenotypes. We have the population, the sex, the hair color, caffeine consumption. And then the genotypes and some related data in the entries. OK, so a GWAS is basically a linear regression for each row or for each variant. And so we've been working to make HALE work very well for genetics data, but to be generic enough that it's not tied to genetics data. So we have a generic method, linear regression rows, that just does a linear regression per row of your matrix table, no matter what the data is. And so we can use that to do the GWAS. And I made, as much as possible, the methods I'm using in this practical, I added links here to go to the HALE documentation. So I want to give you some sort of outline of where to find more information on these things in the documentation. So linear regression rows is a method. And so you can find that in the outline over here. And so the methods are so these are the things you call on like tables or matrix tables. So one of the arguments here, or all these arguments are fields on the matrix table. And I also forgot, I also wanted to point out, so another section of our documentation is the how-to guides. And this would be a good next step after this tutorial. So you can go find the thing you're most interested in and have like a more focused walkthrough on different aspects of HALE. OK, so we're going to do a GWAS on the caffeine consumption phenotype. So in our linear regression, y, or the dependent variable, is caffeine consumption. X, the independent variable, is the number of alternate alleles. And then by adding the 1.0 as a covariate, we get that we're also fitting a y-intercept. So if we run that, and then, so the result I'm storing in a variable called GWAS, and that's actually a HALE table. So we can call describe on that and see what we got back. So we got a table that's still keyed by locus and alleles. So we're saying for each variant, we have some information. So the beta here is the slope of the linear regression. And then we have some measures like p-value of the strength of the association. And so now we have this table. Oops. This table we call GWAS. So let's use some plotting to get a visualization of the results. So the standard plot for GWAS results is the Manhattan plot. And we have that built in to our plotting library. And so we're going to put the p-value field into the Manhattan plot. And see if I might need to zoom out to really see this. So what we get is a plot where the horizontal axis is loci on the genome. And the vertical axis is the negative log of the p-value. So the higher the dot, the smaller the p-value, the less likely of seeing that association by chance. But how do we know if this is a reasonable looking Manhattan plot? So another common next step is the QQ plot. And so we're going to draw this, again, looking at the p-values. And this is showing us, so each dot is a variant, and it's showing us the expected quantile of that p-value versus the quantile we actually saw in our results. And so this is showing us that we're seeing these high p-values much more frequently than we would expect. So that's a good sign that there's some confounding variable that we're not accounting for. So a common way of improving that is by finding some way to account for population stratification. Question? Okay. And so we can do that using PCA. So PCA is a way of, it's a generic way of projecting very high dimensional data down onto a small number of dimensions. And it turns out when you do this on genetic data, the small number of dimensions you get out is a good approximation for like ancestry group of the sample. So we have both a generic PCA method, so I linked here. So that's in the statistics section of the methods. And so you can use that on any numeric data. But when you're working with genetic data, there's some common steps you want to do to normalize the data to get reasonable results. And so we have the specialized method, the HWE normalized PCA. So that's the Hardy-Weinberg equilibrium normalized. So we can just run that directly on our genotypes. And we'll ask for the loadings in addition to the scores. So I'll run this. And so you notice this method is giving us back 3 different pieces of information. And if you're new to Python, this is a little syntactic trick of storing the 3 results in 3 variables in a single line. So what are those 3 results we're getting back? So eigenvalues is just an array of doubles. And I'll get in a second to what each of these really means. And then we have scores, which is a table keyed by sample, and the loadings, which is a table keyed by variant. So let's look at each one of these. So the loadings, we'll look at the first few rows here. So this is keyed by variant. And for each variant, we get, so there's 10 numbers, because, so we didn't specify, but by default, PCA is giving you back 10 principal components. So if you look at like each column of these arrays, we're getting so 10 different like weighted sums of all the loci. So the way I think of this is each one of these weighted sums is sort of a new virtual variant we've computed that is a very, it explains a lot of the variation we're seeing across these samples. So we've sort of collapsed this large number of variants down to just 10. And so the scores are sort of the, for each sample, we have that sample's genome at each of these sort of virtual variants. So for each sample, we again get an array of 10 numbers, which we can think of as either this sort of collapsed genome or as a point in some low-dimensional space. And in the third practical, we'll actually look at some of those plots of looking at these principal components as coordinates. And then finally, the eigenvalues is just for each of those 10 principal components, we get a number saying sort of how strongly does that principal component explain the variation in our data. So what we want to do with this now, so we're mostly interested in the scores. So for each sample, we have these 10 principal components. And we want to add those as covariates to our linear regression because we think of these 10 numbers as being a good signal for what was the background ancestry of that sample. So we want to take our matrix table and add in those principal components as part of the data in the matrix table. So annotate calls is going to add a new column field called PCA. And we're going to look up, for each sample in our matrix table, we're going to look up the corresponding row in the PCA scores table and add its data to this column field. And we don't have it in here, but let me now add in a describe. And I don't remember, if we haven't seen it before, here's describe without the widget. It comes up a little faster. If you're on a command line, this is the only thing you can do. And so it's just a text printout of what are all the fields on this matrix table. And so we can see in the column fields, we've added a PCA struct which contains an array of the scores for that sample. OK, so now we want to run the GWAS again, so do another linear regression for each row, where the only difference is we've added to the covariates. So we've added both the first 3 principal components, and we've also added the sex. So if we run that again and then print the QQ plot, we can see if it looks any different. And so this looks a lot better, right? At the small, more common p-values, it's much closer to the expected line. It's still not great, but we've definitely made an improvement. And then if we also redraw the Manhattan plot, you can see that right, the bulk of the variants are much further below the significance line. And we're still seeing maybe one hit. OK, anybody have any questions before I move on to the second part?
So why this improved QQ plot is not looking as good as you would like? Because of the size of the sample we have, or because only of 10 principal components we included?
That's a good question. I don't have a great answer. There's some confounding variable we haven't accounted for. Does anybody have?
This is also like pretend simulated data. So like we simulated it like with some randomness in it. There's not actually any real. Like we made up this correlation by simulating it, so it's just never going to look exactly the same as like it would look from like real, like a real phenotype. Any other questions?
Quick question: how do you know you are not overly correcting because you picked the top 3 pieces? Like how do you know how many pieces to pick, and how do you know if you're over adjusting for the structure and you're not removing actual relevant structure in the population?
Can you repeat the question?
How do you know how many PCs to use? How do you know you're not overcorrecting? Again, I'm not a geneticist, but my feeling is.
How do you know how many PCs to use?
Okay, well, most of the time you use the first 2, sorry. It depends on what you're adjusting for. Like if you're adjusting for ancestry, one of the key rule of thumbs is always use the first 3 PCs because that usually explains the most differences. Another thing you could do is you could plot your PCs out and see when there's a plateau. So that would actually know when your PCs no longer explain your differences, and then you could just insert first 10 PCs, for instance.
But the issue is that maybe some of those pieces are not correlated with ancestry, but some biologically relevant factors that you're also correcting for.
So in that case, if you are assuming that your PCs may have some confounding towards its explanation, there would be other things you could adjust in your model as well. So if you think there's a biological difference outside of your PCs, that you need to adjust for. That would be an additional term in your model that you need to insert.
The opposite way. That is to say, you are saying you can add confounding factors beside pieces. What I'm saying is that pieces themselves may be of biological relevance that you're removing, right?
True. So if that's the case, if you knowing very well, because there's not like, as far as I know, there isn't like a way per se to look at whether your PCs only explain your ancestries or not. If you want to be safer, I think that was one of the main reasons like when we learned in grad school, like always use the first 3 PCs, because you're probably going to have other differences which are going to be washed out in your model. So rule of thumb from like grad school 4 years ago, first 3 PCs. If any other person would like to chime in, because like that's my experience with PCs. If any other person thinks there's a different way of adjusting it, seriously, love to hear it. Sorry, but that's the best that I can. Thank you.
Thank you.
No worries.
Any other questions?
Okay, so in part 1, we immediately after we imported the data, we filtered out the rare variants. So in the second part, we're going to do the opposite. So let's import the data, and this time filter down to just the variants that only appear in less than 1% of the population. And so let's look at, so we mentioned that there's not enough data there for a linear regression to be very effective. So what else can we do? So one idea is if these variants are rare, then maybe if we sum some of them together, we'll get enough data to see some association. And so we can look for variants that are all in the same gene and assume that variations in the same gene might be contributing to the same phenotype. And so that way we get to look at some of Hale's methods for doing grouping and aggregations within groups. So we'll start by re-importing the same data, and then we do this same filter where the only difference is we changed from greater than to less than here. So now we're looking at just the rare variants. And now, so we want to look at variants that occur in the same gene. So we need to get some information on genes. So we have this table of gene annotations that we're going to import. And then let's look at what's in this. So we have a table with just 5 fields. Yeah.
A few steps ago, we filtered for a little frequency.
Mm-hmm.
Weren't we taking out the ones that are rare, less than 1%?
So filter says keep the things where this is true.
Oh, keep them. Okay.
Yeah.
If you were to switch it to like the other way, then you remove all the rays.
Right.
So the text says remove. I think that's why it's confusing. The text says we'll remove the variant.
Oh, it does.
That's why it's confusing.
That is confusing.
Thank you for noticing that too.
Yeah. We should fix that before we share it. Oh, that reminds me. Somebody on the text asked, can we download the tutorials later? And we had a plan for making them available,
So
right?
what I will do is I'll send the tarball containing the scripts, the slides, and if you need any other information, just send me a Slack. I think it's Kumar Virban or something, and I'll be there for you. Like, or on Zoom, yeah, like a Friends commercial. 3 people laughed.
OK. So we've imported this table of gene annotations. And it has just 5 fields. So there's a gene ID and a gene name. And then for each gene, the chromosome and the start and end position. And so we want to incorporate this into our variant table. And we can also ask how many genes are in this table. So there's about 56,000. OK, so the main step here is this new method called transmute. And so again, I included a link to the documentation. So transmute is, this time I'm looking in the documentation for the table class, because transmute is a method that you can call on a table. So this is kind of like annotate. Annotate just adds new fields. Transmute is useful in the situation where you want to take like a few existing fields and replace them by some combination. So the idea is inside of transmute, I reference the fields chromosome start and end. So anything I reference is going to be removed, and the new interval field is going to be added. And so I added describes both before and after, so we can really see what it does. So before transmute, I have chromosome start and end. And then after, those three fields are gone, and I've added the interval field. And so interval, locus interval, is one of the genetics specialized Hale types, which just represents an interval on the genome. And now, so we can take this table, which now has an interval field, and we can make interval now be the key of this table. So behind the scenes, that means we're sorting the data by this interval field. But what it mostly means is now I can treat this keyed gene table as sort of like a Python dictionary where I can look up data by the key. And this is also a little bit tricky because this is a locus, not an interval. So Hale knows about intervals, and it says, if I have a table that's keyed by interval, and I try to look up by a locus, it'll find some interval that contains that locus. So I'm taking my matrix table that we filtered before down to just rare variants, and I'm adding a new row field where I take the locus of that variant, look up what gene it contains it, and add that gene name.
What if you can overlap in genes?
I believe this will randomly pick one of them. Is that right?
Yeah, there's a way to get all of them, all of the matches.
There is, yeah. So yeah, this syntax just gets one of them. I think it's the join function. I won't take time to do that. But yeah, a method called join, which gives you more options for how to combine it. So now if we show what we have, right, so this time I'm just showing the gene field, so I get to see the row keys, the locus and alleles, and then the gene field. And so you can see this looks vaguely right. For each variant, we've annotated it with a gene. OK, so now we want to do the grouping and aggregation. So let's remember what our data looks like again. So we have on the variants, each variant is annotated with a gene. And now, so I'm going to take this matrix table, I'm going to group the rows by gene. So I'm going to collapse rows together that share the same gene. And then across those groups, I'm going to aggregate and compute the number of variants that contain a non-zero number of alternate allele. So what we end up with, so we've grouped rows by gene, which means after this, we have one row per gene instead of one row per variant. And the field, the only field, is going to be this aggregate we computed, the number of variants. So we can run this, and then we do another step where we take this and we filter out those genes, or we filter down to those genes which had a non-zero number of variants across the population. So let's see what this looks like afterwards. So if we look at rows, before we had variant-keyed rows. Now we have gene-keyed rows. So we have one row per gene. And in the entries, we have, for each sample, the number of variants that sample has in that gene. And we can also look at the top few rows of this. In this case, they're all zeros. But that just means each one of these genes, the sample that contains some variants in that gene have just been cut off in this tiny display. OK, so now let's finally do the linear regression. So the idea is we want to keep caffeine consumption as our dependent variable. But this time we're going to take these grouped sums per gene as our independent variable and the same covariates. And Manhattan plots don't work very well for gene-keyed results. So instead we'll just, oops. So let's just print out the top few rows of the result. So we have for each gene, we have some linear regression results. And so here's our p-values. And so we can compare these with the result from the common variant, GWAS. There's mostly pretty low p-values. This one's the only one that's maybe possibly significant. OK, so the couple minutes left on this practical. Anybody have any other questions? OK, great. So we'll break for 10 minutes before the third practical. And all of us will be walking around in case anybody has any questions they want to ask.